Assalamu alaikum. My name is Sibzada Muhammad Humza. My registration number is MB320222011. Today is my presentation. The presentation topic is regulation of gene expressions. Semester M.phil second, subject molecular biology. Instructor Dr. Niaz Muhammad. This is my outlines: gene regulation, appearance, the basic concept, types, negative gene regulation, positive gene regulation. First of all, we discuss about regulation of gene expression. The regulation of gene expression refer to the mechanisms that control the level and timing of gene activity or protein production within the cells. It is the fundamental process in a living organism and play crucial roles in determining cellular function and development and response to the environment. It means that gene can be turned on and off. If we understand how microbial genes are expressed, turned on and off, they can help us to control disease causing bacteria. Those bacteria which cause a disease, if we understand how their gene can be turned on and off, we can control this type of bacteria. So we will discuss the gene regulation of gene expressions in the like operon models. Operons, the basic concept. What is operons? Operon is a cluster of function-related genes can be under coordinated controlled by a single on and off switch. The cluster and functions. There is a structures genes that are known as like A, like Z, like Y. The regulatory switch is a segment of DNA called an operator. Operator usually positioned within the promoter regions. An operon is the entire stretch of DNA that include the operators, the promoters, and the genes that it controls. The repressor protein can be active or inactive depending on the presence of other molecules. When lactose is present, the lac genes are expressed because L-lactose bind to the lac repressor proteins and keep it from binding to the lac operators. We see in the diagrams that if the lactose is absent, the repressor active, operon off. This is the entire stretch of DNA, and this region is known as regulatory genes. This is a promoter regions. This is RNA polymerase bind to the promoter regions. mRNA is this 5 to 3, and this is the proteins, active repressors. If lactose is absent, the active repressor bind to the operator regions, operon off, there is no RNA formation can be made. If lactose is present, repressor inactive, operon is on. This is also entire state of DNA. This is the promoter regions. RNA polymerase bind to the promoter regions. mRNA proteins. This is the inducer L-lactose. If L-lactose is bind to the proteins, they cannot bind to the operator regions. So the RNA formation can be made like Z, like Y, like A. This is the structured genes. Like Z encode beta galactosidase is an intracellular enzymes that cleave the disaccharide lactose into glucose and galactose. Like Y encode a beta galactosidase permease, a membrane bound transport proteins. They pump lactose into the cells, and like A encode beta galactosidase transacetylase is an enzyme that transfer acetyl group from acetyl-CoA and beta galactosidase. If lactose is present, so the repressor protein is inactive and operon is on, the L-lactose inducer is bind to the protein regions and they cannot bind to the operator regions, so that RNA formation can be made. What is negative gene regulations? Negative gene regulation and positive gene regulations. Negative gene regulation have two types: repressible operons and inducible operons. A repressible operon is one that is usually on, binding of a repressor to the operator, and they shut off transcriptions. An inducible operon is one that is usually off molecules. This is called inducer. Has function that inactivate the repressor protein and turn on the transcriptions. If you see in the diagrams, this is the entire stage of DNA. Also now we did it. If tryptophan is absent, so the repressor protein is inactive. This is the repressor protein is inactive, and operon is off. They cannot bind to the operator regions, so the RNA formation can be made. A, B, C, D, and E. This is a polypeptide subunits that make enzyme for tryptophan synthesis. If tryptophan is present, so the repressor protein active and operon is off. This is the entire stage of DNA. Also now we know that is mRNA is the proteins. This is a co-repressors. Is a co-repressor bind to the protein regions. Active. This is known as active repressors, and they cannot bind to the operator regions. So the no RNA formation can be made. Positive gene regulation. Some operon are also subject to positive controls through a stimulator protein known as CAP. Catabolic activator proteins as function is inactivator for transcriptions. If CAP is activated by binding with cyclic AMP, so the activated CAP catabolic activator protein attached to the promoter region for the lac operon. Its function is increase the affinity of RNA polymerase, thus accelerating transcriptions. When the glucose level increase, CAP detaches from the lac operons and transcription returns to normal rate. If we see in the diagrams, the CAP, this is inactive CAP. This is a CAP is bind to the inactive CAP. This is active CAP. This region is CAP active binding site. This is RNA polymerase bind to the foreign transcriptions. So lactose present, so the glucose is scarce, say MP level high, and abundant like mRNA are synthesized. In this diagram we see that if lactose is present, glucose present, say MP level low, so the little like mRNA is synthesized. This is the difference between these two diagrams. There is CMP level is high and this CMP level is low. So that abundant like mRNA is synthesized and that little like mRNA is synthesized. This is known as a positive gene regulations. Thank you.